Hearing will come to order. Good morning. I'd like to welcome everyone to this subcommittee's hearing on metrology, the measurement needs to support the development of biologics and biosimilars. While I'm very, very aware that other policy issues related to biologics and biosimilars are being considered by Congress, today we are focused on the role that we can play in helping develop the underlying science needed to support the growing biologics industry in general. As I have studied the challenges of developing biological drugs, I've realized that some of the issues facing researchers may be addressed through the same paradigm used for traditional pharmacologic drug development, but in other arenas, the measurement tools to completely characterize relevant pharmacologic products do not exist for biologics today. I have learned as a member of this subcommittee, and I have frequently said that if you can't measure it, it doesn't really exist for technologic or economic purposes. It is could be an important item of faith, but it is not an item of economics or technology. I do believe that this is the crux of the inconclusive nature of the biologics debate, this difficulty in characterization and measurement, which is why this subcommittee has convened this hearing. And this is, at least at this point, I view this as the beginning of a series of hearings that we will hold on this and related biologics topics. This is not a new area of inquiry for the Science and Technology Committee. This committee was the first in Congress to hold hearings on the science and potential of other growing scientific fields, such as recombinant DNA, cloning, genome mapping, and genetic testing. The committee's emphasis has always been focused on meeting the metrology needs that allow these new technologies to move forward. I would like to think that the Science and Technology Committee was successful in realizing that goal in a number of other arenas. In that line, today's hearing will focus on the metrology needs of the biologics industry. And this will be the first in a series of hearings surrounding potentially personalized medicine and genetic diagnostics. One additional issue that I want to address today is the interaction between the industry and the federal government to date. I welcome the suggestions of our industry witnesses on how the relationship between NIST and industry might be enhanced to ensure that NIST can fully anticipate the industry's metrology needs. The thrust of these questions will not be to criticize, but to learn how a better working relationship might be created. I want to thank our witnesses for appearing before the subcommittee, and I look forward to your comments and suggestions. And now I would like to recognize my colleague, Representative Smith, for his opening statement.
Thank you, Mr. Chairman, for calling this hearing today on the very important emerging issue of biologic drugs and the associated standards and measurement science necessary to facilitate their continued safe and effective development. On this committee, we regularly review and consider the impact science and technology and related policies have on our lives. Arguably, in no other area has this impact been so direct and profound as in medical science, where dramatic technological advances have lengthened and improved countless lives here in America and throughout the world. At the heart of these advances are the continuous revolutionary innovations of the pharmaceutical industry. We have almost come to take new life-saving drugs for granted, expecting the arrival of new medications to continue quickly without full appreciation of the complicated and sensitive development system. Central to this system, of course, are strong intellectual property protections, without which there would not be incentives to enable the risk-taking and investment of capital necessary to foster new drugs throughout the long scientific development and regulatory approval process. This is especially important with respect to biologics, where the enormous and unique potential to combat major diseases is hindered by the lack of a regulatory pathway for managing intellectual property. To this end, I am pleased to be a sponsor, along with Chairman Wu, of the Pathway for Biosimilars Act, which would provide the intellectual property protections and regulatory clarity necessary for ensuring and accelerating continued advances in biologics. However, we are here this morning to focus on a separate, potentially limiting factor to biologic drug development, the need for measurement science and standards and development to enable and leverage further advances in biologics. The FDA and industry stakeholders have identified significant measurement science needs to support the regulatory approval and manufacturing processes associated with biopharmaceuticals, and we know NIST has world-class measurement science capabilities well suited to this task. While there appears to be a good opportunity to leverage NIST's capabilities to meet these needs, the details of what exactly needs to be done and what the appropriate roles and responsibilities of NIST, FDA, industry, and other stakeholders should be must be carefully considered. These are complicated questions surrounding an incredibly complex issue. That is, of course, why we are here today, and I certainly hope and expect this hearing provides us a better understanding to this end. I want to welcome the witnesses here today. Thank you for your time out of your busy schedules, and I look forward to a productive discussion.
Thank you very much. If there are any other members who wish to submit additional opening statements, your statements will be added to the record at this point. I would like to introduce our witnesses. Dr. Anthony Mire-Sluis, did I get that right? Very good, is the Executive Director of Global Product Quality and Quality Compliance at Amgen. Dr. Patrick Vink is the Senior Vice President and Global Head of Biologics at Milan GmbH. Right? Dr. Stephen Kozlowski is the Director of the Office of Biotechnology Products and the Office of Pharmaceutical Science at the Center for Drug Evaluation and Research at the United States Food and Drug Administration. We're just going to call you czar of something. Huh? And our final witness is Dr. Willie May, who is the Director of the Chemical Science and Technology Laboratory at the National Institute of Standards and Technology. You will each have 5 minutes for your spoken testimony. Your written testimony will be included in the record in their entirety, and when you complete all of your testimony, we will begin with questions, and each member will have 5 minutes to question the panel. Dr. Mire-Sluis, please begin.
Chairman Wu, Ranking Member Smith, and members of the subcommittee, I'd like to thank you for the opportunity to testify to you today. I have devoted much of my career as a scientific researcher and regulator to the question of how best to standardize and improve methods for biotechnology medicinal products. So I'm particularly grateful for the chance to weigh in on this topic. There is a clear and pressing need for standards and methods to better understand biotechnology medicines and their manufacturing processes. First, although we need standards, they should be the best standards, not just any standards. We also need to understand that even the best standards can and must evolve as science evolves. And finally, while having the best standards possible is necessary, it is not sufficient to assure safety and efficacy. Randomized clinical trials will be needed in order to understand biotechnology medicines the best that we can. First, let us look at the impact of standards on patient safety. One place where it is very critical to have the best and most modern standards is when detecting and measuring whether and how a patient's immune system is reacting to a biological product. That is immunogenicity testing. Immunogenicity happens when your body attacks the medicine that it has been given. Consequences can be that the drug does not work, or even worse, can result in severe side effects. In testing immunogenicity, every company uses different tests and internal standards, which have different capabilities. Because of this, we cannot compare the results that these tests produce. Standardization would allow scientists and clinicians to accurately and consistently measure the immune response against biotechnology products, essentially allowing us to speak the same language. Second, let us look at the impact that standards could have on testing biotechnology products themselves. It is essential that we understand the structure of our biological products and its impact on safety and efficacy, and having the very best standard methods and reference materials available will help us to achieve this. They could also lead to reduced costs by minimizing wasted time and effort and could facilitate greater review efficiencies at the FDA. But measurement standards alone cannot ensure the continued health of the biotech pipeline. It is essential that we preserve the incentives that drive innovative research and development, and that we have a strong science-based FDA. Companies must invest, on average, $1.25 billion to develop and test a biological product. And only 7% of biotech medicines that enter development ever reach the market. That is why strong protection of intellectual property, both patents and data, must remain the cornerstone of this research-intensive, innovation-driven industry. In addition, maintaining the FDA as a world-class, science-driven regulatory agency is essential to public health and safety. Only vigilant government oversight can sustain confidence in the safety and effectiveness of biotechnology products taken by millions of patients. Federal appropriations for FDA have increased in recent years. However, more needs to be done to support the agency's ability to recruit and retain the best and brightest scientists and medical reviewers, modernize the agency's information technology systems, and enhance FDA's scientific capacity. We commend the Science and Technology Committee and this subcommittee for your roles in passing the Competes Act, which has provided a firm foundation for American scientific innovation. Over the past 3 decades, biotechnology products have revolutionized the war against chronic and life-threatening disease. The biotechnology industry, the FDA, and most of all, patients are counting on policymakers to continue to foster biotechnology as our best hope against the devastating diseases that face us today. So thank you for inviting me to testify today, and I'll be pleased to answer any questions you may have.
Thank you very much. Dr. Vink, please proceed.
Good morning. Thank you, Chairman Wu, Ranking Member Smith, and the members of the Subcommittee on Technology and Innovation. My name is Patrick Vink, and I'm the head of Global Biologics at Milan. I'm privileged today to testify before the Subcommittee on behalf of Milan, which over almost half a century has established a solid reputation of manufacturing high-quality, affordable pharmaceuticals. Mylan is the largest US-based generic pharmaceutical manufacturer, with 1 out of every 13 prescriptions dispensed in the US, brand name or generic, being a Mylan product. Today, Mr. Chairman, on the 25th anniversary of Hatch-Waxman, we face a situation comparable to that of 1984, when perpetual monopolies enjoyed by biologics regulated under the PHS Act. Unlike Europe, the US lacks a biosimilar pathway. A viable biosimilar pathway does not require a competitor to re-establish de novo the safety and efficacy of a legacy molecule. Instead, a biosimilars pathway recognizes how much is already known about legacy biologics and enables both regulators and competing biologics manufacturers to appropriately rely upon the prior knowledge and regulatory conclusion flowing from that data. Specifically, this information is the safety and efficacy of the underlying molecule itself. It's that demonstration of comparability, Mr. Chairman, where biologics reference standards could play a crucial role. Comparability is an established scientific and regulatory principle that the branded biopharma industry itself developed with FDA in 1996 to alleviate regulatory burden on the branded industry when they changed the manufacturing process for biologics. An example of this is the product Avonex, which paved the way for biosimilars and, in many important respects, effectively constituted the first biosimilar, because its approval dispelled the age-old paradigm of the product is the process, and established a new biologics regulatory paradigm premised on comparability. As a result of subsequent regulatory developments, comparability was adopted globally as the sameness standard for all biologics. And yet, every time a brand biologic manufacturer has implemented a manufacturing change, that change has resulted in a change in its biologic. The evolutionary process of this comparability creep among branded biologics means there are a number of brand biologics on the market today that may have drifted significantly, or to a minor extent, away from the original versions of those biologics initially approved by the FDA. across the entire lineage of a brand biologic, there is therefore a continuum of substitutability determinations that have maintained the market acceptance and enhanced the abundant market success of so many high-priced biologics in the US market today. It is time to recognize the implications of that regulatory history, accept the scientific conclusions and regulatory confidence it supports, and proceed to apply all logical inferences across the regulatory framework for all biologics going forward. This is where reference biologic standards come in. With the availability of appropriate reference standards, it should be readily ascertainable just how much a branded biologic has drifted between its original approval and FDA's approval of its most recent manufacturing change. Originally approved biologic and the most recent changed biologic enable a fair and readily adoptable set of parameters. These essentially could serve as the regulatory goalposts for approval of a generic biologic. Thus, to be approved, a competing biologic manufacturer would need to demonstrate comparability within that range. From Milan's perspective, a viable approach for this subcommittee is to appropriately incentivize reference standards by creatively linking them in a straightforward manner to existing and future incentives benefiting brand biologics so as to provide a return to American taxpayers, the US healthcare system, and patients in need of these biologics. Specifically, we believe reference standards should be linked directly to these incentives, including any exclusivity, if any. While Milan, like other key stakeholders, is very troubled by the excessive exclusivity period that is currently contemplated, we have identified a constructive way to leverage such exclusivity, if there needs to be any. This can be accomplished by simply conditioning a brand's biotech company's receipt of exclusivity on the brand's voluntary provision of a reference monograph and reference standard materials consisting of supplies of active ingredient and the various iterations of finished product approved by the FDA as comparable. The monograph would be published as the reference materials are evaluated and sold on a not-for-profit basis to companies and researchers for analytical testing purposes. NIST certainly would be an appropriate repository for such reference standard materials. NIST could apply its in-house expertise and develop new analytical tools for regulators and biologic developers in characterizing those reference standards, albeit without developing new standards or guidance, which would become quite problematic at the regulatory interface with FDA. Authorizing NIST to implement such a system could put the US back in a leadership position and enable the US to begin catching up with Europe and other countries that are now many years ahead in terms of enabling patients access to generic biologics. With state-of-the-art analytical methods now available to biologics competitors like Mylan, the operation of a reference standard system would further enhance the global nature of comparability and contribute to a single universal set of tools by which FDA could assess comparability going forward. Such a system would benefit all biologic stakeholders. The approach is suitable and appropriate. I'm convinced about that. Readily implementable and can enhance both the quality and efficiency of all biologics R&D while enhancing patients' access to biologics that can help save lives. In closing, Mr. Chairman, I again want to thank you and the subcommittee on behalf of Milan for this opportunity to present our perspective on the critical importance of establishing a biologics reference standard system, as Milan has proposed. Towards that end, Mr. Chairman, Milan looks forward to working with the subcommittee to implement this approach, and I welcome the opportunity to address your questions.
Thank you very much. Dr. Kozlowski.
Good morning, Chairman Wu, Ranking Member Smith, and members of the subcommittee. I am Dr. Stephen Kozlowski, Director of the Office of Biotechnology and the Center for Drug Evaluation and Research in the FDA. Thank you for this opportunity to discuss how the development of measurement science, standards, and related technologies might make it easier to understand the composition of FDA-regulated biological products and the benefits that could be gained from these advances. The term biological product or biologic includes products that have been manufactured using a biological process, such as a cell line with altered DNA to produce a monoclonal antibody. There are different types of biologics presently on the market, but I will focus on one type today, therapeutic proteins. As part of the FDA's responsibility of ensuring the safety and effectiveness of drugs and biologics sold in the United States, it is important that we be able to understand or to characterize the composition of these products. We want to know what materials they are made up of and how the materials are arranged at a molecular level. That is, what is the molecular structure? I will begin with a general description of biologics with a focus of therapeutic proteins and explain why they are so difficult to characterize. I will then discuss potential benefits that could follow from improved analytical methods and measurement standards. Please take a look at the slide on the displays. This is a graphic representation to scale of a single molecule of the drug aspirin and a single molecule of the protein product human growth hormone. You can see the relative size and complexity. But in comparison to other biologics, human growth hormone is actually simple and well characterized. I was initially going to show a graphic comparing aspirin to a monoclonal antibody, which is 5 times the molecular size of human growth hormone, but then the aspirin would have been rather difficult to even see. I would like to point out 3 specific limitations of our current analytical methods. First, there are additional components not shown on this graphic that we call post-translational modifications. For monoclonal antibodies and many other proteins, these modifications include sugar chains of various sizes, and our current analytical methods are not sufficient to fully assess these additions. Second, we are unable to fully characterize the 3-dimensional structure of biologics. And third, we currently lack methods to measure and quantify the aggregation or the clumping together of protein molecules. I will now turn to 3 specific benefits we might see from improved analytical methods and measurement standards. Improved analytical methods would enable quicker and more confident assessments of the potential effects of manufacturing changes in process, equipment, or raw materials. This could reduce the requirements for animal or human studies for evaluating these manufacturing changes. In addition, for products that have abbreviated pathways for approval, improved analytical methods could facilitate comparison of products and detection of differences between different manufacturers. Number 2, the development of analytical methods would evaluate the quality of a biologic throughout the manufacturing process, and that could provide a superior system for ensuring product quality in the manufacture of all biologics. Improved analytical methods would increase general knowledge in the field of biopharmaceuticals. The FDA can use this knowledge from improved analytical methods to inform our regulatory decisions, and industry can use this knowledge to design even better products. With the development of new analytical methods comes the need for new standards to evaluate them. The term standard can apply to measurements or processes, and although process standards are valuable in ensuring effective manufacturing process operation and validation, today I will focus on measurement standards. A measurement standard can be a standardized test or standardized materials used to evaluate the performance of a measurement method. Standardized test materials can be used to evaluate the precision and accuracy of many different types of analytical technologies, and thus are more likely to foster competition and development of new and improved analytical methods by industry and academia. The development of such measurement standards would also be extremely valuable for ensuring both current and future methods are working properly and provide consistent results from assay to assay and from laboratory to laboratory. In conclusion, the field of biopharmaceuticals is advancing rapidly, in many ways more rapidly than analytical technologies. We have identified 3 specific properties of biologics that we cannot sufficiently measure but are very important to medicinal activity: post-translational modifications, 3-dimensional structure, and protein aggregation. Furthermore, reliable and discriminating material standards would enhance use of current technologies and encourage new technologies to fill current gaps. Thank you for the opportunity to testify today. I am happy to address any questions you may have.
Thank you, Dr. Kozlowski. Dr. May, please proceed.
Good morning, Chairman Wu, Ranking Member Smith, and members of the subcommittee. Thank you for the invitation to testify today. I am Willie May, Director of the Chemical Science and Technology Laboratory at the National Institute of Standards and Technology. Additionally, for the past several years, I've led a strategic planning effort for NIST program growth in the biosciences. The previous speakers have discussed the need for additional measurement science and measurement standards to improve the quality and efficiency in the development, manufacture, and regulatory approval of biologic drugs. Therefore, I will focus my comments on our past experiences in successfully responding to other health-related measurement problems and our capabilities for addressing the measurement and standards needs associated with biologic drugs. We have used our expertise in measurement science and standards to address important problems in healthcare since the 1920s. Over the years, our capabilities and our programs have expanded and evolved in accordance with both societal and industry needs. The primary focus of our current program in healthcare is on the provision of reference methods and human serum-based standards for clinical diagnostics and on standards for medical imaging. In both these areas, NIST traceable measurement standards and calibrations are reducing misdiagnoses, wasteful repeat testing, and treatment decisions based on inaccurate measurement results. NIST can also make critical contributions to underpin the development and the regulatory approval process for biologic drugs. NIST brings to the table a unique combination of expertise in the physical, chemical, and biological measurement sciences. These, along with our expertise in statistics and information science, provide us with the tools required to support a more accurate assessment of the sameness of biologic drugs made by different manufacturers and/or by differing manufacturing processes, improved safety and efficacy, and improved efficiency and reliability in manufacturing processes. Based on extensive discussions with our colleagues at FDA and the pharmaceutical industry, we've identified 5 critical areas where improved measurement methods and standards would benefit both FDA and companies that produce innovative as well as generic biologic drugs. First, the assessment of structural sameness. In this area, NIST expertise in the determination of protein structure and function and protein measurement science could be used to develop quality assurance standards for the measurement methods used to compare post-translational modifications and three-dimensional structure. In predicting adverse immune response in patients, in addition to developing reference methods and standards for protein aggregation, our expertise in protein measurement science and cell system science can be expanded and applied to support a better understanding of, first, the protein aggregation process, and secondly, its induction of adverse human responses to biologic drugs. Developing a comprehensive understanding of the inner complex workings of production cells, in this area, NIST expertise can enable a better understanding of the genetics and complex biochemical networks of cells used in bioreactors. This would support industry efforts to optimize the production of drugs with desired features, namely low immunogenicity and the appropriate post-translational modifications and three-dimensional structure to facilitate efficacy. Predicting drug function and toxicity. NIST expertise in cellular and protein measurement science, genetic testing, and bioinformatics could be used to support a more accurate characterizations of the human cell types most often used in toxicity assays. This would in turn support development of more accurate measurement systems and modeling tools for predicting therapeutic function and adverse human reactions to candidate drugs. And finally, contamination from the manufacturing process and packaging. In this area, NIST expertise in analytical chemistry and protein chemistry can provide the reference methods and quality assurance standards for measurements used to detect and quantify potential contaminants such as unwanted proteins from production cells, viruses, metals, and various organic compounds. NIST has already begun to act on some of these needs. We've started a pilot effort focused on improved measurement methods and standards for glycosylation and aggregation. However, NIST, and I'm sure my colleagues from FDA and industry would agree that there is much more to be done. We at NIST will continue our outreach to stakeholders and determine and refine our best path forward for addressing the critical measurement and standards challenges associated with biologic drugs. So in summary, measurement science and measurement standards for biologic drugs would facilitate scientifically sound and fact-based decision-making in research and development, manufacturing, and the regulatory approval process for biologics. Mr. Chairman, thank you for this opportunity to testify today. This completes my statement, and I, too, will be happy to answer questions.
I thank the panel, each and every witness. We are now going to start questions from the panel, and each member will have 5 minutes to ask questions. And I will begin with myself. It's been a while since I've been exposed to biochemistry, so please bear with me. When you all talk about biologics and biosimilars and measurement and what you can measure and what the need areas are, if you will, Dr. Kozlowski, you seem to list a couple of areas where we need work, and I think that was implicit or explicit in each of your testimony. Are you saying that that we can, well, that what we have is the translation process, but that post-translation, whether it's glycosylation or aggregation or the folding into 3D structures, that beyond the translation stage, there's shall we say, a whole lot of wiggle in what the same translational product ultimately becomes?
There have been a lot of advances to date in being able to characterize molecules, and we do know a lot about post-translational modifications with cutting-edge technologies, but we don't know everything. There are areas where we're lacking the capabilities. Some of these capabilities are more in academic labs and not necessarily translatable as well to industry routine use. So I think we're much better at knowing the very primary structure, the list of amino acids and sequence in a protein, but our ability to account for all the different ways they're modified is lacking. But that's not to say we can't do it at all. And I think that, in terms of the molecules that I showed in my slide, human growth hormone was approved through an abbreviated pathway, both in the U.S. and Europe, through the 505 pathway, and so we felt we knew enough from the characterization of that molecule to make some judgment about an abbreviated pathway. So I think we have a lot of capabilities now. The question is how to make them better, because there's still a lot of uncertainty and gaps in those areas.
So we're a lot better at the translational end, and improvements would be helpful in the other arenas?
Yes.
Okay. Thank you for that. Purely a curiosity question, I guess, although hopefully it'll be helpful to my understanding going forward. I'd like to ask you about each of your companies' interactions with NIST, your perceptions of NIST's attempts to engage the biotech industry, what's been done well, what can be improved.
So I can speak from my personal experience and Amgen's experience of working with NIST on this program through the work that I have done at NIBSC and the World Health Organization. I'm very well versed in the mechanisms for producing standard methods and reference standards. I've collaborated very closely with NIST on this effort, providing advice from an industry perspective as to our priorities. Obviously, there are multiple different areas that we could explore from a scientific perspective, and it's naive to think that we can do them all in one go. There has to be some form of prioritization. From our perspective, I think the protection of patient safety obviously rises to the top of the list. So the issues, for instance, of immunogenicity and linking structure to those possible potential side effects, I think is of increased relevance. I would also say that from a perspective of having worked specifically for institutes whose role is to improve methods and standards directly related to personal health, that I think transparency is a big requirement for any institute working in that manner. These methods and standards cannot be provided in a vacuum. There has to be the highest scientific rigor associated with these methods, as described in our testimony. Making a bad standard does not do anybody any good. And therefore, sharing industry and research expertise, I think, is vital. So I feel that outreach from NIST maybe should be increased throughout the industry as well as the scientific community.
Thank you very much, Dr. Mire-Sluis. Dr. Vink.
Yes, Mylan, as a generics manufacturer, has interacted on several occasions with NIST, mainly also via the work for the U.S. Pharmacopoeia and other areas. Being now also entering into biologics as a generic manufacturer, we see a big role for NIST, as was laid down in my testimony. We see a big opportunity of laying down standards and making objective comparability tools available so that everybody is held to the same standards. NIST can play a very big role in creating that transparency, creating standards that are applicable for everyone, and we completely agree with what is said in the table here. Every progress we make is another step in better understanding biologics. We come a long way in the past 15 years. Every step, especially for our area, biosimilars, will further help better characterizing these drugs, reducing the burden of clinical trials, which are currently still necessary there.
Thank you very much. My time has expired. Dr. May, since I've invited the other witnesses to comment about how NIST, what things NIST can do, when it comes back to my turn, I plan to ask you about your views of how Congress can enable you to do your job better. And with that, Mr. Smith, 5 minutes.
Thank you. Dr. Kozlowski, could you tell us approximately how much funding currently is spent at FDA on biologics research and what the research really focuses on?
So I'm not really prepared to provide the exact funding for what happens with biologics at the FDA. I can tell you that there are laboratories within the Office of Biotechnology Product which look at the manufacturing of biologics, including characterization. And we are in discussion, in fact, with NIST on moving forward on some of those projects together. We also look at biological assays that measure the activity of molecules, which is another way of characterize them. And there are other labs within the Center for Drugs Evaluation and Research which characterize proteins using current methods and may actually look at some samples that are provided for them. So we have capabilities. Again, you know, could we do more with more capabilities? That's always true.
Could you speak to or share with us if you're comfortable that there's not a lot of overlap between NIST and FDA, but yet still working together? I mean, that's sometimes a very delicate balance, but both Dr. Kozlowski and Dr. May.
So I think overlap is always a tricky question. It is a value to have some core capabilities in an organization simply so that they can communicate and work together. And so some level of overlap in technology is good. I think, you know, you need to be communicating, and that's the way to leverage the least overlap in big ways and to get the most benefit. And for instance, the FDA and NIST had a meeting a number of years ago on the issue of characterizing proteins that they co-sponsored and invited industry, and I think that was a great opportunity for dialogue and further meetings like this that involve both FDA, NIST, and industry together may be good ways of figuring out what our overlaps are, how to work best together, and how to do things in a way that as a combined group makes the most progress.
Okay. As you know, NIST has absolutely no regulatory authority. We're not lead agency on anything except measurements, and we focus on measurement science, technology, and standards that have impact across other areas where other agencies in the federal government do have lead agency responsibility. So the only way for us to be successful is to collaborate with both the industry and other federal agencies in areas where they have interest in lead agency responsibility. And we do the things that we do and do well, that is the measurement science, the underpinning measurement science, technology, and the provision of standards.
Okay, thank you. And Dr. Vink, in your testimony, you propose a repository within NIST, federal government basically, that would then sell some of the information to other companies testing and research. And given the nature of biologic drugs, do you think that such an arrangement might undermine the intellectual property, the facets of intellectual property and certainly the incentives to move forward in the future?
No, not per se. We believe that holding such a repository over there is actually part of what we all need to know as a service to public health. This is the standard that we hold every product which is approved to. So we believe this is more a tool to guarantee for public health that there is a standard for every product and that everybody who wants to compare itself to that standard can be measured in an objective, transparent way. And the IP is guaranteed by the IP legislation which is in place. This will only allow regulatory authorities to hold the current product comparable to the standard. B, once the patent is expired, it will open up everyone who wants to make a similar product available and be measured to that standard. We don't believe that there is a IP issue around it per se.
Okay, thank you. Thank you, Mr. Chairman.
Mr. Lujan. Whoops, Mr. Lujan has slipped away to vote, perhaps. Ms. Biggert.
Thank you, Mr. Chairman, and thank all of you for being here. I'd like to just follow up on what Mr. Smith was talking about and find out what the other members of the panel think about that. And before that, I just wanted to ask one question. I think in your materials, Dr. Vink, you had a case, Burlex Laboratory versus the FDA. Was that part of your testimony? Or I was concerned about the standards and the conclusion of the case where the FDA allowed a new drug to go on the market, which was never really tested, but it was a test of a similar drug that allowed that. So I'd like to know, as far as the intellectual property from the other members, but also, is that the way our regulations work, that a drug really doesn't have to go through? And what it says was FDA did not act unlawfully when it determined that Avonex is clinically superior to Bederson, and to approve Avonex for use by patients with MS without requiring clinical trials of Avonex, and 3, issued its comparative guidance document without notice and comment rulemaking. Dr. Sluis, could you comment on that?
To comment on the Avonex experience, or
well, on that, and also the intellectual property question, I think that Dr. Vink raises for allowing other companies to have to test somebody else's drug even before it's on the market.
So I think as far as the intellectual property issue goes, it's all down to the timing. Is that this is actually not a unique experience for the biotechnology industry. The pharmacopoeias have been supplying reference standards and monographs for products for many years. And Amgen itself is collaborating with both the European Pharmacopoeia and the US Pharmacopoeia to provide just that, reference material and the description of the tests we use for our products.
Okay.
So I think this is nothing unusual. As I say, it's just a matter of timing. What we don't want to do is to lose the intellectual property that allows for innovation. I mean, the innovative industry is the industry that is producing new and novel drugs to benefit our patients.
Do you think that we are losing the innovation creativity? I know, again, Dr. Vink's testimony talked about the fact that we are behind the European countries and so many other countries in our development of drugs.
I'm not sure that that perspective is entirely correct, in the sense that in Europe the regulations for the pathway for biosimilars is somewhat more advanced than it is in the US. I think the standards that have been created in Europe are those necessary to maintain the safety and efficacy for patients, and do include the requirement for clinical studies for biosimilar products. So I think it's a matter of that the US now has to develop similar regulations that focus on patient safety, product efficacy, whilst retaining those incentives that are not going to damage the innovative work that the innovator industry does, or we will lose the chance to create new and novel medicines for our patients.
Dr. Kozlowski, could you comment on that?
So I think there is no technological advantage that Europe has in considering these products. So again, I think that there are technological issues for everybody in terms of better methods to facilitate, how much clinical information is necessary, but I don't think there's actually a scientific advantage. I think it's a question of what pathways are available for what types of products.
And Dr. Vink, maybe comment, since you're the one that raised the issue.
Commenting on the last part where I did not yet comment on what is the difference between the different areas of the world, which was actually the last part. I think, as Dr. Kozlowski and also Dr. Mire-Sluis said, the difference is that there is a pathway. We believe that the pathway that is actually in vigor in Europe is working well. It leaves scientific discretion with the regulatory authority. Not every molecule is the same. And every time, and that is also why we support so much NIST, the better we characterize the drug, the more we can shift the balance from actual characterization of the drug to that part of establishing the sameness and reducing the burden of unnecessary clinical confirmatory trials. The better you know what you're talking about, the less that is needed. But one thing is clear, safety of patients is at the foremost important thing for everybody in the industry.
Okay. Thank you. I'll yield back.
Thank you. We have 2 votes called, and there are approximately 6 minutes. That's NBA time, 6 minutes before the clock runs out. It's my intention to try to get to everyone's questions and then adjourn the hearing if possible. And if not, I'll ask the witnesses to kindly stay until we can return. Dr. May, I promise to give you a shot at how Congress can do a better job. So other than sending NIST more money, what are some legislative improvements that would help NIST do its job better?
Certainly providing legislation that authorized more research in general on supported research in general on the 5 critical areas that I mentioned, and that those funds need not necessarily come to NIST, but certainly there needs to be a lot of work on measurement science to understand the underlying mechanisms and phenomena that are associated with things like aggregation and many of the phenomena that we've all said are critical to the regulatory approval of biologic drugs. Obviously, we would certainly be happy to see you support any budget initiatives that come from the executive branch in this area.
Thank you, Dr. May. Dr. Vink, Milan, you try to do, or you do, biosimilars. Since a pathway exists in Europe and currently either does not exist or is a very narrow pathway here in the United States, is your biosimilars activity primarily in Europe, and then what, you're looking at the pipeline in the United States? Just trying to understand your Mylan's business.
Milan has recently entered the area of biosimilars. After the integration of 2 companies, we became a global company. And our activity is a global one. Our effort is a global one. We believe that the biosimilar scientific standards are the same, are very much aligned between the different continents. So we aim at global product files and a global strategy. And we do believe that the US will also offer a tremendous opportunity for patients, healthcare, and companies to enter the area of biosimilars. So, of course, currently the market for us is open in Europe and has recently opened in Japan. We have a strong belief that this will be also soon in the US. So we do not make any difference for regions with respect to our strategy.
But your activity is a little bit higher in those other areas right now, and part of it is a biosimilars pathway, but further research and reference materials and metrology would assist in those efforts.
Absolutely.
Mr. Smith, any questions? Well, we have a number of other questions. This is the nature of this institution, that you make a plan and then you adjust upon contact with reality. I thank the witnesses. We would like to submit further questions in writing, and perhaps you and the organizations that you represent would be kind enough to respond. And with that, again, I want to thank each and every one of you for coming here, for testifying, and we will adjourn this hearing. Thank you very much.